Okay, hello and welcome to the ZOOMinar presentations today. I also would like to welcome Professor Melachini to the panel from McMaster University. And today we have Dr. Martina Pannuzzo from Italy and Dr. Irving Vega from Michigan State University. First speaker will be Dr. Martina, and she'll be introduced by Dr. Melachini.
Thank you. Thank you, Rams. It's my pleasure to introduce Dr. Martina Pannuzzo, who actually started her career with a laurea from the University of Catania in Italy. That's also the place where she pursued her PhD. And with her PhD, then she went for a postdoctoral fellowship at the University of Erlangen in Nuremberg, where she stayed a few years. And after that, she followed up with another postdoctoral fellowship at Carnegie Mellon in the US. And then she was recruited back on the other side of the Atlantic, back in Italy, with a Marie Curie fellowship at the Italian Institute of Technology, where she stayed a few years. And then she was recruited to the private sector, I believe a Korean company called PharmCAD, for whom she's working now, but she's based in Sicily, I understand. So Martina is joining us right now from Sicily, and their talk is about a combined theoretical and computational approach to disentangle the molecular events at the intersection of amyloid, pore, and fibril formation. Martina, we look forward to your talk. Without further ado, the floor is yours.
Thank you. Thank you very much for the introduction, and thank you for giving me the opportunity to present this work to the major experts in the field. And I think my contribution today would be more about opening questions, but I hope in the end this might stimulate some debate, especially considering that it seems that we are pretty far from finding a successful therapeutic treatment for these diseases. So it would be, I think it's time to reconsider our most consolidated beliefs. So in my talk today, I will present some theoretical and computational data on the molecular events at the intersection of amyloid, pore, and fibrils formation, where the interactions of amyloid peptides with the lipid environment might play as a watershed. And it's quite surprising to me the fact that despite decades of research in this field, we still, and besides, the lack of understanding of how these amyloid peptides are involved in the pathogenic mechanism of this disease, we still don't fully understand their physiological function. And it's clear that if we understand their physiological, their role, then it might be better to understand what can cause their dysfunction and the eventual appearance of amyloid biomarkers, and eventually try to repair and prevent the damage in a more precise way. So for this reason, probably the structure of my talk will sound a bit unconventional, but I would like to present in the first part of the talk, I would like to provide some biological context to these amyloid peptides, at least in my understanding. And I'll make specifically the case of amyloid beta. And in the second part, instead, I will try to, in the second part, I will try instead to discuss the data in relation to both the physiological or pathological relevance that this might have. And in the end, I would like to propose a couple of hypotheses that it would be nice to test, at least for me, to test experimentally. So when we talk about amyloidosis, this This is probably the first image that comes to our mind regarding the amyloid aggregation pathway, starting from soluble monomers in solution that will assemble and forms these oligomers, structured oligomers that further assemble in fibrils and in the end in insoluble mature plaques that are rich in cross beta structures. And it's quite accepted nowadays that the toxicity correlates with the appearance of these oligomers, whereas the plaque the appearance of the plaque is probably related to the progression, is a consequence of the progression of the disease. And it's clear how this picture becomes much more complex if we try to contextualize the biological environment where we have an infinite number of potential partners that can bind amyloid peptides and change completely the kinetics and aggregation of these interactions. Starting from the interaction with the membrane that is believed to be at the basis of toxicity. And the general idea we know that is that these oligomers are believed to seems to disrupt the membrane integrity. And the most accredited hypothesis is that pre-structures oligomers enter into the membrane and form pores that will provoke ion dishomeostasis. Now, considering that the self-assembling of these peptides is mediated by the formation of beta sheets, these pores are believed to be beta barrels. But we should also keep in mind that amyloid oligomers, and I'll specifically talk about Ebitda, have shown to have also some physiological function. For example, they've been shown to protect from infectious diseases, to repair leakages in the blood-brain barrier, to promote recovery from injury, and to regulate synaptic function. Specifically regarding the modulation of synaptic function, it has been shown experimentally that in physiological condition at picomolar concentration, Ebitda can positively modulate synaptic plasticity and memory. Whereas at nanomolar concentration, Ebitda might participate in a negative feedback that could keep neuronal hyperactivity in check. Now, these authors, interestingly, have proposed that Ebitda might physiologically protect neurons from damage upon hyperexcitation. But how Ebitda can do that? So this mechanism that I'm going to describe has been experimentally observed, but it has been proposed to explain the pathogenic mechanism of Ebitda. But in principle, this mechanism might work as well to explain the effect of Ebitda in physiological concentration when in protecting the neurons from damage and upon hyperexcitation. So the general idea is that long-term potentiation, that is a form of synaptic plasticity involved probably in learning and memory, induces rapid synaptogenesis that is mediated by the release of neurotransmitters from synaptic vesicles. Now, upon hyperexcitation, it has been seen that these Ebitda42 forms oligomers that correlate with an increase of cytosolic calcium that activate this CAMK4 that in turn inhibit synaptic vesicle transport. So in this way, new synapses can't be formed and neurons are protected, and Ebitda might indirectly modulate, downregulate neuronal hyperactivity. The idea that Ebitda is involved in synaptic function modulation is further corroborated by the observation that altered Ebitda expression correlate with synaptic dysfunction. And we see this in Down syndrome, where long-term potentiation is impaired, but we have an overexpression of Ebitda. Whereas in the case of autumn spectrum disorders, long-term potentiation is enhanced and we have a low endogenous expression of Ebitda. Now, the high concentration and the prolonged exposure to such high concentration cannot explain the insurgence of Alzheimer's. Otherwise, young people with Down syndrome will have Alzheimer's. But the co-occurrence of aging and mutation-dependent alteration might provoke the accumulation of Ebitda and eventually provoke the insurgence of Alzheimer's disease. So what I want to underline here is the concept that when we experimentally perform an experiment within a certain window of concentration of Ebitda, or we take the measurements in a specific time, we might observe some effects that we might attribute to Ebitda as potential harming, but might instead correlate with the synaptic, might have instead, a physiological implication. Now, in any case, the action of Ebitda involves the interaction with the membrane, and it's clear experimentally that amyloid peptides strongly interact with the membrane and they can spontaneously form pores. What we don't know is how we go from the production of Ebitda that happens in the endosomal membrane to the appearance of embedded oligomers in the plasma membrane. Now, we know that these experiments are really difficult to perform for several reasons, because of the chameleonic nature of these peptides that tend to change configuration depending on the binding partner, because we have a low peptide endogenous concentration, because these peptides are highly prone to aggregate because they have some hydrophobicity. And also the problem is also that these aggregates that they can form have shape and morphology that can change completely depending on the environment where they form. So in the end, we don't know what's the Ebitda configuration, what's the aggregation state of these peptides when they interact with the membrane. We don't know what's the pore structure. So these steps might be investigated using a theoretical approach and molecular dynamics simulations. This is a technique that allows to reproduce what atoms do in real life. So atoms move because they feel forces. So if we give enough time to the system, this will tendentially explore all possible configuration. So it's like having a computational microscope. Basically, in the end, from microscopic information, we can derive microscopic observables that tell us something about the properties of the system. And as it happens in experiments, we might change between different levels of resolution. So using high accuracy models, we want to look into, like in the interactions between residues or the folding of the protein, where the chemistry is well described, whereas we will use a lower resolution models if we want to investigate like collective phenomena typical of viscoelastic systems, like, I don't know, membrane deformation of the aggregation of peptides inside the membrane, where that allows to speed up the simulation, basically. So the chemistry details are not fully there, but the model is accurate enough to catch this kind of phenomena that are mostly physics driven in this time and length scales. And the good thing is that we can always go back and forth between different levels of accuracy, depending on the details we want to include and we want to look into in our study. Now, going back to the beginning, let's say, we know that Ebitda is produced from the sequential cleavage of beta and gamma secretases of the amyloid precursor. And we know that Ebitda has a certain degree of hydrophobicity. So the first question would be to me to try to understand how these peptides leaves the membrane in the first place, and how does these peptides travel. We have some, there are some experimental evidences that can give some hints about these aspects. For example, the fact that we know that ApoE binds Ebitda in vitro and in vivo, and my work as a chaperone and a carrier in Ebitda trafficking. There are computer simulations that have predicted that Ebitda monomers in helical configuration might be stable inside a membrane in the presence of an asymmetric distribution of cholesterol at physiological concentration. This is the typical composition of a healthy membrane. Whereas in the absence of cholesterol or weak cholesterol that is symmetrically distributed, that would mimic more the composition of a membrane in aged condition. In this case, the peptide seems to shift at the interface. The other interesting observation is that Ebitda, the monomeric state, can interact with glycolipids and reinsert inside the membrane, probably coadjuvated by the presence of cholesterol that tend to co-localize with glycolipids. And at a low Ebitda GM1 ratio, this peptide assumes preferentially an helical configuration. We have evidenced recently that if there are free lipids in solution, Ebitda might bind to these lipids and form the complex at the specific ratio 1:1 that has a higher propensity to reinsert inside the membrane as compared to the bare protein or the protein bound to many lipids, two more lipids, because the hydrophobicity of this complex is overall higher as compared to the other two cases. Another interesting observation is that the binding of the Ebitda peptide to these lipids promotes the formation of helical structures, whereas in the absence of lipid, these peptides will tendentially form beta sheets. And another important observation from my co-authors is that if we have many free lipids in solution, these peptides will preferentially form pores inside the membrane, whereas if we have few lipids in solution, these peptides will tendentially form fibrils. So it's like lipids compete for the self-assembling of these peptides and promote the insertion of Ebitda inside the membrane, of this amyloid peptide inside the membrane. Now, this observation has important implications in the pathogenic perspective, because we know that there is a lipid dyshomeostasis correlate with these amyloid diseases. And also it's interesting to know that ApoE mutations are a risk factor for Alzheimer's. In particular, ApoE mutated has a less binding affinity for Ebitda. So in this case, what might happen is that if a ApoE, for example, cannot sequester, cannot bind Ebitda, Ebitda might be sequestered instead by free lipids, and this might promote the reinsertion of Ebitda in a specific way in an off-target membrane. But the idea is that in general, we might presume that these monomers of Ebitda might exist inside the membrane in a helical configuration. So the question is try to understand what happens if we have many of them. So this, in this study, we follow the self-assembling of these peptides in a pure POPC membrane and in a POPC membrane in presence of cholesterol symmetrically or asymmetrically distributed. So the first interesting result is that in any case, these peptides tend to self-assemble. And I think this is an interesting result because we are used to think that the self-assembling of Ebitda peptides proceeds through the formation of beta sheets. But we clearly see that here peptides in helical configuration are still equally highly prone to aggregate. The other interesting observation is that if we are in the absence of cholesterol, these peptides form aggregates with an elongated shape. Whereas if we have the cholesterol, they will preferentially form round-shaped aggregates, because cholesterol has the ability to stabilize the boundary of these aggregates. If we zoom in the structure of these aggregates, we clearly could see some chiral order. So we want to explain this using a theoretical approach. So the idea is that if we have a charged ribbon in bulk solution, this will tendentially fold as a helical or a twisted ribbon, because the exposure of the charged groups, of the charges groups to the solvent is maximized in this way, whereas the intra-repulsion between the charges is minimized. If we now embed this ribbon inside a membrane, the problem is that here the ribbon cannot fully twist, because it will mean for the charged groups to go across the hydrophobic core, which is energetically is forbidden. So in the end, they will form this kind of frustrated helix. The other complication when we talk about amyloid peptides is that the charges are mainly distributed in one of the two terminals, at the N-terminal. So what happens is that this end part will tend to twist, and they will laterally push the neighbor lipids, whereas the C-terminal, that is basically neutral, will not affect or perturb the membrane, the bottom layer's integrity. So in the end, there is a different increase in the surface area of the two layers of this membrane that will induce spontaneous curvature in the membrane. So the results we observed in simulations was that these aggregates are able to stimulate membrane curvature. So we wanted to understand better this interplay between curvature and aggregation, but we noticed that if we were using a bilayer stabilized by periodic boundary condition that makes these layers infinite over the 3 dimension, this simulation box we call basically inhibits the membrane deformation. And the other problem is that the local curvature is hidden by fluctuation. So we instead use an open patch. This open patch will spontaneously close to form a full vesicle. And here the barrier of this transition will depend on the line tension that here is stabilized by these short lipids on the edge, on the bending rigidity, meaning how soft, how stiff is this membrane, which is an intrinsic properties depending on the composition of the membrane, and on the bending power of impurities. So we are amplifying the response of this patch to the presence of inclusions that might induce some curvature stress. The interesting result, we may focus just on the black profile that corresponds to the case with a higher concentration of peptides, is that the full black profile is the case where the peptides are embedded in a flat bilayers. Here the aggregate size grows over time. If we instead have the peptides embedded inside a patch that can bend, the interesting result is that once they reach a critical threshold forming in size where this aggregate basically consists of 3 or 4 peptides, the membrane vesiculates. But the interesting aspect is that the aggregation stops. So the vesiculation, the formation, the curvature has an effect in preventing further aggregation and stabilize oligomers of smaller size. Now, if we add now cholesterol to the membrane that is stabilized by periodic boundary conditions, so the flat bilayers, but we now add cholesterol symmetrically or asymmetrically distributed as a mimic for aged or healthy conditions, what we see is that in the case of an asymmetric distribution of cholesterol, as these peptides aggregate, they will induce a strong vesiculation. Now, if we restrain the membrane, so if we don't allow the membrane to bend, what happens is that Also, in the case of an asymmetric distribution of cholesterol, the cluster grows. But as this reaches a critical size that before was inducing membrane vesiculation, now is extruded out from the membrane. So it's like a way for the membrane to release the stress, because the membrane now that cannot bend, as a way to release the stress, will just push out these clusters that will continually now will tend to grow. So the concept that I want to stress here, I don't want to jump to any conclusion and say this is healthy or unhealthy, and this correlates with an aging condition, so on, because we know that membranes are a complex system with many of lipid components that might counterbalance in a different ways. The concept that I want to stress here is that when we design experiments, I believe we should properly account for the ability of the membrane to bend, because there is membranes are soft system and they can respond to the interactions and the stress induced by the interaction with inclusion. And we clearly, this is because we clearly see that there is a dynamic interplay between aggregation and curvature, because as these peptides aggregate, the membrane vesiculate, but as this membrane bends, it slows down aggregation. So we might follow in some artifacts when we study the interaction of peptides, for example, with a supported bilayers that cannot bend, or if we study the interaction with a preformed vesicle. And the other point that I want to stress is that here I'm studying the self-assembling of peptides that are already embedded inside the membrane. Whereas experimentally, when it studied the effect of curvature on the aggregation, we are talking about peptides that are isogenous. They are added to the preformed vesicles from the outside. So the structures that these oligomers might have in solution might be completely different from the structures they form when we start from peptides that are embedded inside the membrane. And I want to show this because this is a zoom of the aggregate, a small aggregate that is stabilized inside the vesicle, where we can see clearly a tetramer that has a round shape and that will remind the shape of a pore. And the interesting aspect is that these peptides here have a helical configuration. So here we form basically a helical pore. Whereas probably if we start with peptide in solution that will already self-assemble and form a shift structure, and probably they will enter inside the membrane, probably will be more embedded barrier. So the idea is that what we see in the end might tightly, strongly depend on the initial configuration of these peptides that will depend on the native environment in the first place. So in conclusions, alpha helical monomers might be the most representative configuration under certain conditions, like in lipid environment. Inside the APOPC membranes, these peptides self-assemble to form aggregates with a frustrated helix configuration that have an elongated shape in the absence of cholesterol, a round shape in the presence of cholesterol. Tetramers are the most representative building blocks in the formation of these supramolecular assemblies. The aggregates stress membrane curvature, but in turn, membrane curvature slows down aggregation and stabilize smaller aggregates. And I think this is important to account properly in setting experiments. And finally, I would like to conclude, as I anticipated, advancing to crazy hypotheses based on the observation, on the previous observation. So the first one is the fact that we clearly see that Ebitda has a from simulation, that Ebitda has the propensity to stimulate membrane vesiculation. So I wonder, I think it would be interesting to test if Ebitda might mediate its own trafficking, considering that it's known that Ebitda is uptaken inside the cell, but we don't know how it travels. And it's interesting, but we know for sure that it's not mediated by cluttering. But it happens at the raft, at the level of raft, where there are glycolipids and cholesterol. So it would be interesting to see if Ebitda is able to modulate its own uptake or its own trafficking. The other hypothesis is that we know that amyloid peptides form pores. We know that these pores, I mean, it's quite clearly, it's quite evident that these pores have a high affinity for calcium. That is a second messenger really important in biology. And it would be, I think, interesting to test if these pores might have a physiological function. Might be these pores important, for example, to mediate a physiological concentration that responds to hyperexcitation. For example, might be these pores directly correlate with the increase of calcium, cytosolic calcium that goes to activate the CAMK4. Because in principle, these pores might form, promote the increase of cytosolic calcium, and then as they accumulate, they will induce vesiculations and the reuptake of Ebitda and the depletion of Ebitda, the plasma membrane, that will restore the signaling. With aging, the membrane might become stiffer because we know that there is an increase of cholesterol and that becomes symmetrical, and this might affect the mechanical properties of the membrane that might have hard time now to vesiculate. And as a potential response, these pores might form clusters, aggregates, and then be extruded from the membrane and further grow in solution. In perspective, I would like to mention the fact that we might take advantage in understanding the physiological role of Ebitda. We might take advantage of the existence of other diseases where the expression of Ebitda is altered, because we might also try to understand better not only Alzheimer's but also other pathologies as well, such as Down syndromes and autism spectrum disorders. So with this, I'd like to end, but I want to acknowledge my co-authors, Professor Audino, that unfortunately passed away last years, and we miss him a lot. Professor La Rosa, that is very active in the field with this group of young scientists, and Dr. Milardi, Professor Cartoon, and Rainer Bockmann, that have contributed to some of the work that I presented today. Thank you for your attention, and I will be happy to debate the validity of this statement with you, and I'll be happy to get your question. Thank you.
Thank you, Martina, for a very stimulating talk. You already have 3 questions. First one is from Dr. Sikun Liu. Nice talk. What do you think is the transmembrane region for Ebitda 42?
So the transmembrane region might change depending on the composition of the membrane. Now, there seems to go deeper is the presence of cholesterol, and partially then terminal sweet move translate at the interface in the absence of cholesterol. This is what we see in simulation using a pure POPC membrane or a membrane with cholesterol.
Okay, thank you, Martina. You also have a couple of questions from Rams. Are the Ebitda pores ion or charge selective? How do they vary with the lipid composition?
So in simulations, I've performed, once I performed some studies on this is really a bit difficult to study in simulations because depends, I mean, I don't always get, as you saw before, I have a standard average size of these oligomers, but I'm not sure if these, sometimes I get tetramers, sometimes I get pentamers. And I performed some studies on the flux of waters and ions across, specifically calcium ions, and they can go, the flux can vary depending on the lipid composition. Especially this was strange because in the presence of cholesterol, these pores look very much permeable, in contrast to the case where these pores were in the absence of cholesterol. So the lipid composition has an impact in the permeability of the pores that I observed or I've studied. But of course, for this, I think we need to use much more different techniques, much more accurate. This was just studies just to look at the flux of waters and the flux of ions, but I cannot say this is
sure
like that. Or...
Thank you, Martina. One more question from Rams. How does a pre-existing membrane curvature affect the A-beta oligomerization?
Yeah,
you touched on that a little bit in your talk.
This is an interesting question because, I mean, if we start with the peptides inside the vesicle, I haven't tried this, but I think it's an interesting question.
Okay.
So I haven't studied this. I know that once these oligomers form, then they are stable in this vesicle once it's closed. So they will not further aggregate. But I haven't studied from monomers inside a preformed vesicle because I was interested to look at the dynamic interplay between the two, how the aggregate size would have affected the membrane bending and the other way around.
Thank you, Martina. You have one more question from Dr. Becker about cholesterol, concerning the importance of cholesterol for membrane plasticity. Could there be an important link between the use of statins and Alzheimer's disease?
I don't have idea. I'm not a doctor. Sorry, I cannot answer this question. Yeah, I think it's interesting. Yeah.
I also had a question. I mean, much of your work is under the umbrella of the pore hypothesis, but in the initial part of your talk, you also mentioned the carpeting and detergent hypothesis. Do those, to any extent, apply to the A-beta, or the primary effect of A-beta on membranes is through the formation of pores?
Are you talking from a pathogenic mechanism?
Yeah.
So of course, these are effects that have been observed for the pathogenic mechanism of A-beta. But I mean, the literature, you can find everything and the opposite of everything. So yeah, in principle, physiology depends on the experimental setup, is I think a lot. Okay. On the concentration and of the membrane composition, so many factors.
Sure, it's very complex.
Yes.
Thank you so much, Martina.
Yeah,
please join me in thanking Martina again for a wonderful presentation. Thank you.
Thank you, Martina. It was a nice talk. Thanks, Malachini. Our next speaker is Dr. Irving Vega from Michigan State University, Grand Rapids. Here's a brief introduction to Dr. Vega. Dr. Vega obtained his undergraduate degree in biology from the University of Puerto Rico. His PhD research training was in the Department of Cell Biology and Neuroscience at the Graduate School of New Brunswick, Rutgers University. He was a postdoctoral fellow in the Neuroscience Department at Mayo Clinic, Jacksonville, where he developed his research career focusing on the pathology of Alzheimer's disease. Dr. Vega joined the faculty as an associate professor in the Department of Translational Neuroscience at Michigan State University College of Human Medicine campus in Grand Rapids in 2014. His current research focuses on molecular and biochemical mechanisms that modulate the accumulation of pathological tau proteins in Alzheimer's disease and related dementia. I would like to welcome Dr. Vega. Dr. Vega, the floor is yours. You need to unmute yourself.
Okay.
Yeah.
Yes. Okay, perfect. Can you hear me well now? Yeah.
Yes. Yes.
Good. Thank you for the invitation, Ram, and Martina, a great presentation. I really learned a lot from your presentation, so thank you for that. Today, I'm gonna change a little bit the topic to tau protein aggregation. It seems to be correlated to Abeta, but I'm gonna talk mostly about proteins that a particular protein that we found modulates tau protein aggregation. So why is not? Ooh, okay, hold on a second. It's frozen. Okay. Okay, so in terms of conflict of interest, I don't have financial conflict of interest to declare. So as Martina was mentioning, in Alzheimer's disease, there is the aggregation of Abeta 42, and the most prevalent hypothesis is the amyloid cascade hypothesis that indicates that there is a correlation between Abeta aggregation and the aggregation with tau proteins in the intracellular compartment of the cells, generating oligomers, filaments, and then the neurofibrillar tangles that are observed in the brain of people that endure Alzheimer's disease for 5 to 20 years in some cases. So the general consensus is that the Abeta seems like to be an inducer, and tau mediates the neuronal death of the cells, especially the neurons. In the case of tau, the data accumulated throughout the years indicates that there is a range of toxicity of the different molecules or different structures that are formed once tau become aberrantly modified, or there's other protein-protein interactions that mediate the transition from monomeric to dimers, trimers, oligomers, filaments, and then the tangles that we observe as the final ultrastructure that tau can form. There are many proteins that have been shown to associate or regulate these transitions in the structure. So from the monomeric to the dimer and trimer, there's chaperone proteins known to modulate that dynamic structure of tau and actually regulate its function either as a microtubule associated protein or other functions in signaling and more that continue to be highlighted in the literature, and seems to be regulated by phosphorylation and other post-translational modification, including truncation. If the proteins continue, tau proteins continue to form in dimer or trimers, there is pathways that become activated to degradation, mainly by the proteasome pathway, but also there's some evidence of lysosomal autophagosomal degradation of the tau protein. There's once these dimers are formed, there is some proteins that associate with it, like HB90 or FKBP51, that together they actually promote the formation of oligomeric forms. The definition of oligomeric form in the tau field is still unclear. Whether what is the constitution of an oligomer for tau. But it seems the data to hold that there is in this oligomeric form, the tau protein can be released from the cell. Still, the pathway is still unclear how the oligomeric form of tau can be released from the cell and perhaps uptake by neighboring cells. And that pathway of uptake is still unknown as well. And that's what is referred to the transmissibility of the molecule. So the oligomeric form has been shown to be uptake by neurons in vitro and also in brain using mouse model. And it's been to be associated with toxic form, which can serve as a seed to promote further aggregation of the tau, intrinsic tau in the neurons. As the protein continue to form continue to aggregate in the oligomeric form that leads to filaments. And there are proteins that actually fight the formation of filaments, apparently, like TIA1, that actually try to maintain a tau in this form, in this oligomeric form, instead of the filament. And again, this oligomeric, when you compare oligomeric versus filament, those structures like pair helical filaments that are the ones observed in Alzheimer's disease, the transmissibility of the filament is reduced. So the toxicity is assumed to be reduced to, because the oligomeric are the ones more prone to induce that aggregation in the neighboring neuron, like serving as a seed. Additionally, once they go to filaments, they can then collapse in this structure that we call neurofibrillary tangles. And this ultrastructure is a collection of these filaments that come together. And we know that tangle-bearing neurons are still functional. And when we look at a brain that endured Alzheimer's for years, the remaining neurons have those tangles. So, and one more piece of data is that when you have tangle-like structures and you do these seed experiments to look at the toxicity, neurons cannot uptake tangles. They can uptake filaments and they can uptake oligomers, but not tangle form. So it seems that these tangles evolve as a neuroprotective mechanisms that sequester this toxic tau in the neurons and prevent to go outside of the cells. But that's a hypothesis right now. So we were interested to find proteins associated with tau in the course of the formation of those aggregated form. And we use an animal model that expressed the human tau protein with a mutation found in frontotemporal dementia. Is the P301l mutation. And this animal model was developed by Yara Lewis and Mike Hutton in Mayo Clinic. And these animals, one phenotype is that in an age-dependent manner, they develop motor impairment because the tau gene is under the prion promoter, which is expressing all neurons, including neurons in the spinal cord. And they develop motor impairment about 9 months to 12 months of age. And also, one other characteristic is that these animals develop aggregated tau, represented here in a Western blot of purified tau through the sarcosyl insoluble preparation. So the P3 fraction here represents the sarcosyl insoluble fraction. And this is a Western blot against the human tau. So the antibody use was specific to the human tau. That's why the non-transgenic animals, there is nothing. But you see that throughout the age, in an age-dependent manner, you see more accumulation of tau protein, mostly aberrantly phosphorylated tau protein in the sarcosyl insoluble fraction. So these animals serve as a model to study the progression of that formation of tau pathology. So we did immunoprecipitation of the tau proteins and then combined that with mass spectrometry. And of course, confirmed that tau was there. We found other proteins in here. I'm just illustrating SS70, differentially associated with protein, but also we noticed very faint bands at that time, just a few years ago, we did this original experiment where this faint band in the bottom of the gel represented a protein known as EFHD2. I'm going to call it as EFN2. And this protein has two EFN motifs and that binds calcium. We demonstrate that it coordinates 2 molecules of calcium per molecule of protein. He has a very disturbed N terminus, and he has a coil domain in the C terminus. So of course, these studies were done in the mouse model. We validated the interaction with tau in humans, in human tissue, postmortem tissue, where we also found that in Alzheimer's disease brain, the abundance of the protein seems to be increased compared to normal aging. And these results have been recently confirmed by independent groups where they have seen that EFN2 abundance of the protein increases in Alzheimer's disease compared to normal aging. We also look at the localization of EFN2 in brain tissue or human brain tissue, and we found that EFN2 co-localized in the somatodendric compartment with tau. In here, we are using PHF1, which is an antibody that recognizes tau in the tangle form, and it was found that they co-localize in the somatodendric compartment where the aggregate is formed. We took sarcosylin-soluble preparation from human brain tissue, and we follow that analysis with gold immunogold electron microscopy to see if EFN2 and pathological tau, again, it's shown here by PHF1, an antibody that recognizes aberrantly phosphorylated tau, and mostly in the aggregated form. And we look at the sarcosyl fraction from human brain, and here the large particles are EFN2 and the small particles are tau, and we found structures that were co-labeled with both immunogold label using electron microscopy after purifying the sarcosylin-soluble fraction from the brain tissue, indicating that EFN2 is aggregated with tau in these tangle structures. So what is the role of EFN2 and how EFN2 can modify tau aggregation? And that was one of the questions that I have the data today to present to you guys, where we were interested to answer the question, in this process from oligomer form to tangle, where EFN2 forms. We know that we look at the last stages where we see the EFN2 co-localizing with tangles, but is that what EFN2 associated with, or EFN2 is associated with tau throughout this process? So we know that tau can form these liquid doublets in liquid-liquid phase separation experiments, and we decided to look at the properties of EFN2 and tau, and whether EFN2 affects the property of the liquid-liquid phase separation of tau. And the first thing is that we found that EFN2 can actually co-localize with tau in these liquid doublets that tau can form. So they are together in the doublets, so they do phase separation. But depending of the presence of absence of calcium, in the presence of calcium, you see that is in these round, nice doublets that actually are very dynamic. When we did a FRAT analysis, fluorescent recovery after photobleaching, we found that in the presence of calcium, that structures in the presence of calcium are very dynamic and they can photo recover. But in the absence of calcium, EFN2 with tau induces these solid-like structures, these aggregates that are completely different to the dynamic doublets that are seen with tau. And that once it's photobleached, it doesn't recover. So it's a solid-like structure in the absence of calcium. So there is the absence of calcium actually promote these solid-like structures where the presence of calcium maintain tau and EFN2 in these dynamic structures. So it seems that EFN2, depending of whether there is calcium or not, it can modulate how the dynamic structure of the tau protein. So we say, wait, that is okay, that it can induce that solid-like structure. We want to go further and study the molecules that are how the structure is formed. So we did some in vitro study again with using electron microscopy. And what we found is that, well, in here, we have EFN2 alone, we have EFN2 without the coil domain, and to short the time, because of the time of the talk, I don't present in the data, but we demonstrated that EFN2 associate with tau through the coil domain. So the coil domain is required to see the association between EFN2 and tau. So with EFN2 alone, we see sort of that formation of filaments, and we have published that EFN2 can self-oligomerize in vitro in a protein-dependent manner, and actually calcium can affect that formation. And that self-oligomerization depends on the coil domain as well. We use a tau filament formation inducer, aragonoic acid. And aragonoic acid is known to take monomeric tau and form these pair helical filaments in vitro. For some reason, in the presence of aragonoic acid, EFN2 without the coil domain can form also filaments. And then you have these filamentous structures that are formed also in the presence of aragonoic acid with EFN2. So we decided then to put together EFN2 at tau to see what kind of structures we can observe. And in the absence of aragonoic acid, we saw that EFN2 and tau together. Now we see these aggregates that are, we call it neurofibrillary tangle-like structures that are very filamentous around, but it has a core where the proteins come together in that very dense core of those aggregates. In the absence of the coil, we don't see that formation. Then we added aragonoic acid, and then in the absence of the coil, we see filaments. Most likely these are tau filaments, and then in the aragonoic acid and tau and EFN2, we see again the formation of those tangle-like structures that are very dense, and they have filaments like coming together. So to confirm whether EFN2 and tau are together in those structures, we went back to immunogold, and we did immunogold analysis of the structures that we are observing, and we saw again small molecules, gold particles are tau, large particles are EFN2, and we saw that there is co-localization of EFN2 and tau in those dense core particles, that core structures that we saw in EM. So we did some experiments. We're continue to do some experiments. We know that right now, if we use this EFN2 tau and aragonoic acid with or without aragonoic acid in cell culture, we recently found that it cannot induce seeding or induce aggregation of cells of tau expressing cells. So we are proposing, in terms of our hypothesis, that EFN2 is a protein that can take tau in the filamentous form and probably induce the formation of tangles in a neuroprotective way. And that's a hypothesis based on the molecular analysis that we have done, that we see this tangle-like structure form once we incubate with EFN2. So in order to continue testing the role of EFN2, we then moved to a genetic approach. And we developed the EFN2 knockout mice, and we crossed that to the transgenic mice that express the tau, the human tau with the P301l mutation. And after doing some back crossing and making sure that the genome was stable, and we have an isogenic colony, we start doing some experiments to determine what was happening in the progression of tau aggregation in the present or absent of EFN2. And we were able to show that in the knockout, we don't have the protein and we have the wild type. So we compare, for this one, we were comparing the wild type versus the homogeneous knockout. And I have to say that the knockout of EFN2 don't affect the development, behaviors. The animals are normal. So we look at motor impairment because that's a phenotype that is observed in these animals originally. So we have non-transgenic that are wild type versus knockout. There's no differences between them. And then we compare them to transgenic animals, meaning expressing the human tau and EFN2, or the transgenic animal expressing human tau without EFN2. And we saw that there is a slightly rapid progression of motor impairment in the animals that have the absence of EFN2 versus the animals that express the wild type. We also conduct nesting experiments. Nesting measure hippocampal activity. We didn't want to do any maze or any other cognitive assessment on this animal memory test because of the motor impairment. Nesting seems to be is better because it's not that dependent of the movement of the animals. So we did nesting and we saw that there's definitely a rapid decay or more decay on the animals that are transgenic in the accent of EFN2 than those animals that are wild type, or meaning they have both genes, both alleles of EFN2. So it seems that in the absence of EFN2, the progression of the phenotype of neurodegeneration in the animal is exacerbated. We look also at using markers of tau pathology, especially those markers that recognize pre-tangle tau formation. And we use 88, which is a specific phospho antibody to tau, and also S422, which actually also recognize an early phosphorylation event that leads to pre-tangle formation. And in the case of the transgenic animals that are knockout or EFN2, we see a more accumulation of those two markers of pre-tangled tau that in the ones that express both alleles of EFN2. So it seems that in the absent of EFN2, you have an accelerated accumulation of those pre-tangled forms of tau. So we are finalizing the characterization of this animal model right now, and looking deeper into different markers for tau pathology to dissect what is the structure of tau that is mainly accumulated in the knockout versus the wild type, and this is done in young animals because in all the animals there might be some tangles too. So we are looking at throughout age to see biochemically and also with immunohistochemistry what are the tau forms that are aggregated in the wild type versus the knockout EFN2 knockout mice. So with that, there is a lot of current and past members in the lab that contribute to the work. And also our collaborators here at MSU and also at University of Michigan that have helped us with the different characterizations of tau and EFN2 associations and the funding that support this work. So ready to questions. Thank you.
Thank you very much, Irving, for a nice talk. Please post your questions or raise your hand to join the panel. I'm going to ask a very naive question. I'm not very familiar with the tau literature. So when you talk about oligomers, I know Rakesh Kayed was a very expert in this area. He's not here today. What kind of oligomers are these? Are these metal bound or lipid bound, or any oligomers that can transition to tangles, or when these selective oligomers are toxic and transition to fibers and tangles? Can you give more insights on that?
Yeah. So for the ones that I'm doing in vitro, the ones that we're looking in vitro is only the recombinant protein purified from bacteria to them together. So there's no lipids in the reactions. For the ones in the tissue, in the cells, apparently that there is no lipids that involve in the formation of these oligomers. However, there's protein-protein interactions that might be regulating the formation of these tau oligomeric forms. However, as I alluded in my presentation, in the field, what is the definition of tau oligomeric form is not clear. It can be between 8 to 20 tau proteins. It's still not clear what is the oligomeric forms. In genetically speaking, anything that is not seen as a long filament, as the ones that I was showing with arachidoic acid, are called oligomers. So yeah. And what people are doing right now is that they're actually using these reporter cells that have a gene, the microtubule binding domain of tau attached to GFP, and they do these seeding experiments where they treat the cells with, they look at and they sonicate these tau aggregated forms in vitro and form these small short filaments that they call it oligomers. And those, when these cells are treated with these short oligomers, then they can induce the aggregation of the reporter protein inside the cells. But inside the cells, I think is more, especially in Alzheimer's disease and other tauopathies, there might be other proteins that are regulating that transition between oligomers and filaments and entangles. Hopefully EFN2 is one. We can convince people that EFN2 is one of them.
Thank you very much. I have another naive question on the difference between fibers and tangles. The tangles, you said, they're kind of dead-end species. They don't reverse back. What is known about the tau protein conformation or structure in fibers versus tangles? What makes it more, quite stable compared to fibers?
Yeah, it's this transition to this beta prit structure, right? So it forms from the helical structure to the beta sheet structures, and that leads to the oligomers and the filaments. And then once they're in those filament structure, the transition is not that clear how they go into these tangles. And that's where I think EFN2 plays a role in promoting these filaments to come together into these tangle structures. What is the structure in there? There's recently experiments shown how these, using cryo-EM, they're looking at the formation of these, particularly the region of the microtubule binding domain that is required to form these filaments, and how they form these structure, these loops that are prone to form these filaments and then later on tangles. How they come together, actually the cryo-EM data suggests that depending on the disease that we're talking about is the conformation that region of the protein gets. Yeah.
Thank you. So there's a question in Q&A from Ashutosh Tiwari. Very nice talk. Have you seen variability in toxicity of tau with the different concentrations of EFHD2?
Yeah, very good question, and that's something that we are I show you data only with the knockout, but we have data also with the heterozygous knockout. And we know that that's the reduction of just knocking one allele leads to a reduction of about half of the amount of EFN2 in the brain. However, the data so far shows that reduction of half is the same as the knockout. So the heterozygous data is very similar to the knockout animal. So what the experiment that we want to do is to actually take the knockout and then use viruses, transduce the animals, and to control the expression of EFN2 in specific brain regions. And that actually will allow us to see if now introducing EFN2, we can actually revert the phenotypes that we see. Interestingly, there was a recent paper that confirmed our data that EFN2 is increasing Alzheimer's disease, and it is linked to a microRNA that apparently is regulating the expression of EFN2. And this microRNA is reduced in Alzheimer's disease. And when it's reduced in Alzheimer's disease, now the expression of the mRNA of EFN2 increases. And now you have more protein. So how EFN2 is regulated physiologically to the function, biological function, and why it needs to be maintained regulated by this microRNA is something that goes to the question, what will happen if you modulate the abundance of EFN2 in the cell? But we don't know.
Great, thank you. So please post your questions or join the panel. We can open up for you to ask questions for both the speakers.
Yeah, it will be interesting. Yeah, go ahead, Rams.
Go ahead, Irving.
Okay, no, it's linking it to Martina's presentation. It will be interesting to see, because we don't know how tau is transmitted from one neuron to the other one, and what could be the effect of these A-beta peptides to the membrane that allows these tau oligomers to leak out of the cells or to affect the permeability of the membrane when these peptides are affecting the structure of the plasma membrane or making these curvatures, how that actually help in the release of pathological tau from the cell, and then uptake in neighboring neurons too. So that would be interesting to see. Yeah, I don't know. I was thinking about that when Martina was presenting.
Also, because I think tau and beta are associated also in the quite associated to the synaptic plasticity, right? Because tau control the long-term depression, whereas beta control long-term potentiation. So they must communicate in a way, no? But sorry, I missed what's the role of calcium? Is calcium critical for the formation of these interactions or no? I didn't.
Yeah, so that's another point, because we're looking at CAMP kinases as well. But in the case of calcium, it's not required for the protein-protein interaction between EFN2 and tau.
Okay.
But it's required, but it affects what structure is formed. So if you add calcium, now you have in the liquid-liquid phase separation, if you add calcium, you have very dynamic doublets that when you do the FRAP analysis, it's completely recovered. But when you add calcium, now the proteins get stuck in the absence of calcium, these proteins get stuck into this solid-like structure that don't recover in FRAP analysis. So after photobleaching, they don't recover. So somehow calcium modulates EFN2 dynamics. We are not sure how calcium will affect the structure of EFN2. What we know is that in the absence of calcium, with tau, they form this solid-like solid-like structures in the liquid-liquid phase separation experiments. We also know that CDK5 phosphorylate EFN2 on a specific site, and that phosphorylation affects calcium binding for EFN2. So we don't know if calcium dysregulation, how to put together calcium dysregulation in the cell, the phosphorylation of EFN2 by CDK5, or the activation of CAMP kinases, because we know now that EFN2 associates with CAMP kinase 2. So we have all these pieces that we're trying to put together, try to design experiments to try to link all this data. But yeah, a lot of loose points that we're trying to connect.
Ashutosh.
Hi, first of all, very nice talks, both of you. And I had a question for Martina. You showed some interesting data on relationship of lipids mixed with Ebitda concentration, and the lipid concentration played a role in either it forms a pore-like structure with some generic aggregates, or it forms a fibril-like structure. So is there a critical ratio of protein to lipid, which is the tipping point in terms of what kind of structure is observed?
So the idea is that both the concentration or high concentration of the peptides or a high concentration of free lipids might move this equilibrium towards the formation of pores.
Okay.
So the specific concentration I can't answer because these are experimental data that I've not performed.
Okay. And due to the nature of the interaction, you have also, or somebody checked for what's the relative toxicity? I know it's one is pore formation, another is fibril, like how they relate to toxicity as well.
No, I think they just check the formation of the pore and the formation of the fibers in that experiments.
Okay.
Thank you for the question.
And Irving, coming to tau, tau is a very fascinating protein because it has now its aggregates and its different structures have been linked in different diseases, from the variability in toxicity as well as different diseases. And any thoughts on any, because we have talked about oligomeric structures, any form of structure that you see predominantly more associated, I would say, in a linear fashion with toxicity versus this variability we see that there can be just minute quantities that can be toxic. So have you seen a structure and toxicity relationship?
Yeah, definitely. Once I think when we capture the oligomeriform of tau, what we call oligomeriforms, I think the techniques that we use to detect that, I think tau was toxic way before that. It appears that this aberrant modification of tau, and depending on the localization of the functions of where tau gets affected, because we thought before that tau was only a microtubule associated proteins that stabilize microtubules. That's it, right? Now, as Martina alluded to, now we know that it's in synaptic plasticity, that is involved in signaling, regulating kinases and phosphatases. We know that it's even in the nuclei. So I think that once tau reach a specific post-translational modifications, and the emphasis is in phosphorylation, but there are other post-translational modifications, the protein structure changed from a very dynamic helical, mostly alpha helix structure, to these beta sheets, and that change from these helical structures to the beta sheets send a signal immediately to the cell. And chaperones and other proteins come to action to see if they can solve the problem. That regenerates a lot of need for energy. And that signal, when that signal of tau modification still is there, more tau molecules start changing, more energy consumption start happening in the cell. And I think the cell respond to form these oligomeric filaments, tangle forms, mainly for protection, to take that energy, that huge energy that is creating this molecule in the cytosol to put that entropy to reduce it. But the signal is still there, and more molecules of tau start generating these structures. At some point, the cell may decide to try to get rid of them, but then that's part of that. Yes, this vicious cycle of taking these molecules. Although there are some people that said that tau maybe is secreted regularly as part of this physiological function as well. So there's all these things happening that we don't understand specifically when the toxicity starts. But I think that the toxicity starts way before oligomerics are formed inside the neurons. And maybe the oligomeric form is actually something that triggers in other neighboring neurons toxicity, but inside the neuron, maybe before that. Honestly, I don't know the exact answer to your question, but
thank you. I think this is a good question, and the body of people we are in, I think this is a long-standing question, but still interesting to learn. So thank you very much.
Yeah, appreciate it.
Thank you, Ashutosh. Thank you, Irving and Martina, for great talks. I think let's close this session. Thank you all very much. See you all. Bye-bye.
Thank you. Bye-bye.
Thank you. Bye.
Bye.